Hello everyone and welcome. My name is Selvi and I am an account manager at Novagene Europe. I will be your host for today's webinar and thank you so much for joining us. I am delighted we have participants from various backgrounds with us today. In this session, we are honored to have Dr. Bilal, who will share insights on advices in genomics approaches to characterize Campylobacter directly from clinical samples. Before that, I'll take just a few moments to introduce Dr. Bilal and our company to you. Let's start. Can you see my screen?
Yeah, all good.
Great. Um, Dr. Bilal has been a research scientist at Quantum Institute Bioscience since 2019, with expertise in microbial genomics and metagenomics focused on Campylobacter jejuni. Dr. Bilal has developed innovative protocols for detecting pathogens from clinical samples and led collaborative research with the NHS and international partners, including USA, Slovenia, and Bangladesh. As both a workshop leader and contributor to international scientific programs, Dr. Bilal actively supports global progress in microbial genomics. Dr. Bilal's research interest focused Campylobacter biology, including survival, biofilm, biofilm formation, and persistence in food systems. They study gut dysbiosis linked to Campylobacter infections using advanced genomics and metagenomics approaches. Dr. Bilal also characterizes Campylobacter features in clinical and environmental contexts through genomics tool and will cross-disciplinary collaboration to strengthen microbial surveillance and public health impact. I like to give a few information about genomics research live. It offers your research global exposure through live webinar and expert Q&A session. It is space to discover and exchange innovations and to collaborate across borders with leading scientists in fields. If you want, you can join us with our next webinars as well. I like to mention about our company. Novagene was founded in 2011 and has been growing rapidly since then. And we continuously developing our portfolio of NGS and third generation sequencing. And today we are present with services in various fields of science, from research, oncology to food and water testing. Our first European sequencing center is located in the UK at Cambridge Science Park. The UK lab is equipped with Illumina technology and our patented intelligent sequencing platform, Falcon. This fully automated system integrates multiple precision instruments to streamline DNA and RNA sequencing workflows, significantly reducing turnaround times and minimizing human error. Falcon-free ensures contaminations-free, high-quality results, enabling us to process large volumes of data efficiently and consistently. So unlock the full potential of our research with our comprehensive sequencing services, from the human genome to transcriptome, microbial genomes, and epigenomes. We deliver cutting-edge insights tailored to your needs. Explore complexities of animal and plant genomes with our advanced platforms, and dive deep into proteomics, understand protein dynamics. Powered by the latest technologies, our services offer precision, speed, and reliability, fueling discoveries and that drive science forward. Join the pioneers benefiting from the world-class genomics and proteomics analysis today. So why you should choose Novagene? Choosing Novagene as your sequencing partners means choosing unwavering quality and reliability, and prioritizing excellence through rigorous quality control at every stage, sample, library, data QC, to ensure your results are accurate and trustworthy. Our growing capacity, boosted by the new German lab and our patent Falcon-free technology, not only enhance data quality but also speeds up turnaround times, running seamlessly 24/7. So, yeah, your Novagene contact is always close by. With local representatives who speak your language, we are here to support you every step of the way. Let us help you achieve your research goals with personalized service and expert guides tailored to your needs. Thank you for your attention. That concludes my part for now. I'm pleased to hand over the session to Dr. Bilal. Dr. Bilal, please take it away. Thank you.
One moment, let me share my screen. Can you see my screen, Sylvie?
Yeah, it's perfect.
Okay, perfect. Thank you. So thank you, Selvi, for the invite. Good morning, everyone. And I can say also good evening to some people on the other side of the world. I am Bilal, a postdoc based in the UK at Quadram Institute Bioscience. And in this webinar, I'll share with you how I use metagenomic sequencing to characterize key Campylobacter attributes from clinical stool samples, and how this can reshape diagnostics and public health surveillance for this leading foodborne pathogen. Before we dive into the core topic, I'd like to give you a brief overview of my scientific background. So my scientific journey started with a PhD in microbiology at the University of Sassari in Italy, where I studied Salmonella genomic in a diversity in Algeria. Then, since I joined QIB, my focus has shifted to Campylobacter, especially its genomics, survival, persistence, and contaminations in various environments. So what is Campylobacter infection? Well, Campylobacter is known to be the leading bacterial cause of gastroenteritis globally, including the UK and Europe. It causes an infection of the gut with common symptoms including diarrhea, abdominal pain, fever, and nausea. While symptoms are usually self-limiting, the infection can lead to severe complications like neurological effects and systemic infections. It is estimated that millions of cases of foodborne illnesses worldwide are caused by Campylobacter contamination each year. This image summarizes how Campylobacter spreads and causes illness. Poultry, especially undercooked chicken, is the most frequent source, but the bacteria can also be picked up from unpasteurized milk, raw meat, or contaminated water, often due to animal waste runoff. People sometimes become infected directly by contact with farm animals or pets, and that carry pets that carry the bacteria. The transmission cycle starts when Campylobacter colonizes the intestine of animals and is shed in their feces, contaminating the environment, our food, or our water supply. When people consume these contaminated products, the bacteria reaches the gut, leading to gastroenteritis. And it's worth mentioning that recognizing these key steps highlights why strict food safety, hygienic animal handling, and water monitoring are vital to stopping infection and protecting public health. And now let's take a closer look at the bacterium itself. Campylobacter is a gram-negative, curved, spiral, or rod-shaped bacterium. It possesses a polar flagellum that gives it its characteristic darting motility. The bacteria are non-spore forming and quite small, measuring from 0.2 to 0.8 micrometers wide and can be up to 5 micrometers long. This bacterium has a fastidious nature. It grows in microaerophilic conditions, a profile presenting unique challenges for clinical laboratory detection. This image is a high-resolution electron micrograph of Campylobacter jejuni. It was taken with our QIB imaging facility team. The bacteria's characteristic spiral or corkscrew shape is clearly visible, along with their long whip-like flagella that provide rapid darting motility. This unique morphology allows Campylobacter to move efficiently through the mucous lining of animal and human intestines, aiding infection. The small size, just fractions of micron wide, makes detection and study this bacteria challenging, especially in clinical samples. So diagnosing Campylobacter isn't straightforward. The infectious dose is low, and it's often a minor part of the stool microbiome, making detection challenging. Currently, our diagnostic methods for Campylobacter have limitations. Culture-based methods are slow and can be difficult, while PCR is faster, but it provides limited information beyond presence or absence. Neither of these methods give us a complete picture of the strain's genetic makeup, which is crucial for public health surveillance and understanding the spread of antibiotic resistance. Historically, in our region of the UK, before 2016, culture was considered the gold standard for diagnosis. However, this changed with the introduction of rapid PCR methods. While rapid PCR offers quick results, providing a simple yes or no for Campylobacter, it comes with limitations. We lose crucial information regarding resistance profile, strain types, and also potential contributions from other Campylobacter species in cases labeled as unknown symptoms. Here in this study, we propose a metagenomic approach that will allow us to claim or to reclaim that discarded genomic data. So in the rest of this presentation, I will walk you through our published study. We assessed metagenomic approaches for Campylobacter diagnostics using short-read sequencing services from Novogene Europe. In this study, first, we aimed to evaluate the sequence quality metrics of Campylobacter using metagenomic analysis, focusing on its implications for both clinical and public health purposes. Second, we aimed to validate the accuracy of metagenomic-derived Campylobacter genomes against those cultured from the same stool samples. And this comparison will help ensure that our metagenomic approach is not only effective, but also reliable. There is also more on how long-read sequencing using Oxford Nanopore technologies can do that too. And we elaborated more on that in the paper. Please refer to it. We performed our study across the Norfolk and Waveney region in the UK, analyzing samples between August 2020 and June 2022. This included samples from 42 different gastroenteritis cases gathered from the NHS Microbiology Diagnostic Lab at Norwich and Norfolk University Hospital. So to simplify the process, when patients presented with gastroenteritis symptoms, stool samples are submitted to the diagnostic lab for rapid PCR screening for enteric pathogens, including 3 species of Campylobacter. I then collected the surplus stool samples and transported it to our lab in QIB for direct metagenomic sequencing and conventional microbiology. Refining the methodology, on the other hand, for this project was a significant undertaking. After extensive optimization, I established 3 distinct workflow approaches, each designed to maximize the accuracy and the resolution of Campylobacter detection and characterization using advanced sequencing services provided by Novogene. First, extraction was performed directly on unfiltered stool samples. Following enzymatic depletion of host DNA, this approach aimed to capture the broadest possible microbial diversity present in the clinical samples. Second, we applied a 0.65 micron syringe filtration step to a subset of samples, effectively reducing background noise and selectively enriching for Campylobacter cells. This optimization aimed to enhance the quality of sequencing reads and improve downstream genomic analysis. Third, conventional culture was employed by direct plating of stool samples, yielding isolated Campylobacter colonies. These cultured isolates served as crucial references for benchmarking and validating the metagenome-derived genomes, or MDGs, generated by shotgun sequencing. Altogether, these workflows enabled robust comparative between direct metagenomics and culture-based methods, laying the foundation for our rigorous evaluation of Campylobacter strain detection in clinical settings. So for our first aim, before conducting any downstream data analysis, FastP was used to filter reads for quality and trim adapters. Then taxonomic abundance was estimated using Kraken2 and Bracken to create a microbiological profile of the samples. In this study, the focus was on filtering Campylobacter reads. So Campylobacter genus reads were extracted and assembled using Schoville. Then we conducted our characterization of E. Campylobacter attributes. For our second aim, the MetaRAP pipeline was used for phylogenetic analysis of Campylobacter. We focused on genome refinement, achieving 87% BASCO completeness. This analysis compared metagenome-derived Campylobacter genomes with isolate genomes from the corresponding stool samples. So now let's dive into the results. What we found was very encouraging. The metagenomic sequencing approach showed excellent concordance with qPCR, and this confirms its reliability as a diagnostic tool. More importantly, it also provided us with rich genomic data, which is something traditional methods cannot do. On the other hand, of 42 samples, culture detected Campylobacter in 73%, qPCR in 97%, while direct sequencing achieved 100% genus identification and 65% species identification. However, while metagenomic sequencing excels at genus-level detection, species-level accuracy is more variable, a key consideration for diagnostics. Next, we assessed read and coverage as quality metrics for Campylobacter species detection. In our analysis, the unfiltered samples showed a species identification success rate of 93% when coverage was equal or higher than 5, and Campylobacter reads were over 12,500 reads. In contrast, the filtered samples achieved a 90% success rate, identifying 9 out of our 10 MDGs in the subset that we tested. The 3 plots illustrate our finding effectively. The first plot on the left shows that both filtered and unfiltered samples have similar total read counts. The second plot highlights that filtered samples yielded more Campylobacter reads as filtration reduces background noise and enriches selectively Campylobacter. The third plot on the right demonstrates generally higher coverage in the filtered samples. So these results underscore the significant benefits of filtering for enhanced Campylobacter detection. And as expected, a higher N50 correlates with increased genome completeness and successful species identification. And that was the case for our study here. So the scatter plot displays the genome assembly size distribution. We found that the success rate increases to 100% when N50 exceeds 10,000 base pair. And this, again, underscores the importance of N50 as a quality metric in Campylobacter metagenomic analysis. Next, we assess genome completeness as quality metric for identifying clinical Campylobacter attributes. Our analysis revealed that when we have 60% completeness, we can achieve 100% species identification across our samples. MLST, however, results showed 73% success in unfiltered genomes and 60% in the filtered ones. And as sample size is low, more numbers are needed to test the significance in this case. Moving on to our second aim, we validated Campylobacter metagenome-derived genomes against isolate genomes. This phylogenetic tree on the left illustrates the relationship between the MDGs and isolates from the same stool samples, with MDGs indicated by the black ring. So when zooming on a specific case shown in green, where genomes from multiple isolates and the corresponding MDG cluster closely together, sharing the same sequence type, which validates again our characterization of Campylobacter in the metagenomes. On the other hand, examining the AMR profiles reveal that while our MDG matches many isolates, some belong to the same class of antibiotics, but have different resistance genes. Continuing on our second aim, we observe different aspects of the phylogenetic tree regarding Campylobacter validation. Focusing on the case highlighted with the red dotted frame, we detected 2 distinct sequence types from the isolates, yet the MDG was unable to assign any ST. And this underscores a challenge when multiple STs are present within the same stool sample. However, when we examine the AMR profile, we find that the MDG matches both isolates with ST48, demonstrating that while the sequence typing was unsuccessful on the MDG, the resistance profile still provide valuable insights into the relationship between the isolates and the metagenomes. So in conclusion, direct metagenomic sequencing of stool samples has proven to be a powerful tool for Campylobacter analysis. First, we validated key attributes of Campylobacter in metagenome-derived genomes against culture results, demonstrating high accuracy. Furthermore, our filtration approach significantly increased the number of Campylobacter reads from the MDGs, enhancing the characterization of key attributes. But importantly, our study shows that direct sequencing addresses the gap in Campylobacter genomics, particularly where traditional PCR assays may fall short in providing crucial clinical insight. This method and finding from this study serve as essential guidance for both epidemiologists and clinicians, facilitating the effective use of metagenomic data in Campylobacter genome analysis. I would just like to mention that this study was Campylobacter targeted metagenomics, but in another study, hopefully I can share it with you in another time. We wanted to see how Campylobacter affects the whole microbiome in the infected patients. And again, I used services from Novogene sequencing on a cohort patients of 60 patients, comparing two groups, PCR Campylobacter positive and PCR Campylobacter negative. Hopefully, I can share this with you soon. So finally, I would like to express my gratitude to the entire team, our collaborators, of course, and especially Novogene for their high-quality sequencing services, which were fundamental for the success of this project. And thank you, and I'm happy to take any questions.
Great. Thank you, Dr. Bilal, for this great presentation. And we are also really happy to support your, research. So now we have some questions. I like to start now. So and so the first question is, what were the main challenges in sequencing Campylobacter directly from stool samples compared to cultured isolators?
This is actually the core of my optimization of the workflow, because when I started years ago, there wasn't actually like a commercialized kit to get like a pure metagenomic DNA from stool samples. So I spent like a great deal of time improving that step to get enough pure DNA to use it as an input and then for sequencing it. So that was the biggest challenge in my whole study. When it comes to genomes of Campylobacter, because usually when you compare, like you extract from pure culture of genome, like the bacteria, it's usually easier because you have less contamination, you have more like pure DNA. So that wasn't a big issue for me in this study. The challenge was more with the metagenomic sequencing. I spent a lot of time improving the DNA extraction method. And for instance, at the beginning, when sequencing my first samples, I had the challenge of the host DNA, which is human DNA, which was present in high percentages in my final output data. And that's not helpful when you have like a really low abundant bacteria as Campylobacter. So because, for example, Campylobacter can be like 0 point something% from the whole microbiome. So you want to get enough depth of sequencing as much as you can. So, and that was challenge number one. I needed to add a host depletion step before even going to DNA extraction. And then when going to DNA extraction, at the beginning, I had the challenge that the quantity of the DNA was really low. And so I had to improve that, how to increase the yield of the DNA and then increase the purity of the DNA. That's even before going to sequencing. I hope that answers the question.
Yeah, thank you. It's great. So thank you very much. We have the another question is yeah, what are the next steps in your research? Will you explore along with metagenomics or host pathogen interactions in more detail?
So actually, if you have a look at the same paper, we have a different, I would call it like a different section of the study where we used long-read sequencing from Oxford Nanopore Technology. And in that project, we specifically used adaptive sequencing and standard sequencing. But that was, I used that on mock stool samples where I spiked the stool samples with different percentages of Campylobacter, and I sequenced the same samples, both standard sequencing and then adaptive sequencing. Just to clarify what is adaptive sequencing. So with Me9 sequencing, you have the chance to tell the computer or the machine while you are uh, at the process of sequencing, you upload like genomes of Campylobacter to the system, and you tell the system or the sequencer enrich for Campylobacter. It's like when you recognize the Campylobacter genomes, allow it to be sequencing, and when you see a different genomes, just reject it and don't sequence it at all. So that happens live. So and in that paper, we saw like there is a slight improvement with adaptive sequencing. It improves the quality metrics that we assess using short read sequencing.
Okay, great. We have more questions. And so, yeah, the question is, were there any notable differences in the Campylobacter strains found in Norfolk and Bovini compared to other UK regions?
So that will be like a different project when we use like genomics to like track the strains that are circulated in the region. But in general, like based on I spent like 4 or 5 years collecting samples from the hospital. So we are, I mean, in the data collection I have, so we are finding similar strains. And for example, like this, I can mention the clonal complex 21, which is known to be the most prevalent. So I have that in my collection. So it's actually a mosaic of sequence types. But I mean, I have to do another project just to study that. So that's something I didn't do.
Okay, great. So we have more questions, and is from yeah, it's from Rincon Christian. Given that accuracy was affected by depth, how the optimal sequencing depth is defined. So yeah, it has a big impact on the costs. I think, yeah, the
Yeah,
question is about sequencing depths
I mean,
and
yeah, I mean, I haven't mentioned the depth. In this project, I used 10 gigabytes per sample, and that's, I mean, usually that's enough. So, but if also the cost, because once you go deeper, you pay more. So, if you ask
thank
like interested in a specific one sample, maybe you can go even higher. So,
yeah, thank you.
you're welcome.
And so another question from Celil Özaslan, Büşra Gülay. So, yeah, first of all, thank you very much for your presentation. The question is, we stored the Campylobacter isolates and we cultured in pits at minus 80 degrees Celsius. But when we wanted to grow them again, we found that they didn't grow. What's your recommendation?
I mean, that's not surprising at all. That's Campylobacter. It has like a really fastidious nature in the lab. So if I mean, it should grow technically. So make sure like when you store it and make your stock, you have enough cells. Because I get it. I worked with different pathogens, like Salmonella, for example. You just have a tiny bit and you store it with glycerol and survives forever and it doesn't get much affected to the thaw and freezing cycle. But Campylobacter seems to be really sensitive. So first, I would say when you are making your stock, make sure like you have enough cells, like vortex it really well as well, and then if even if that happens, it could be like a more sensitive strain. It depends, but if that happens, try to enrich it as well before growing it. And I usually grow it also in at 37 degrees. I mean, you could also use 42 degrees. And yeah, so I mean, that's Campylobacter. You know.
Okay, thank you. And the other question from Edisu, and the question is, there was higher detection rate in the filtered sample than unfiltered one, if not mistaken. Is there any justification how it happens?
Yeah, that's actually a good question. So the idea is like the stool samples is really rich in microorganisms. So you have a lot of microorganisms, and Campylobacter usually is like a tiny part of that microbiome. I can tell you, like it's usually 0.3%. Like 1% is really high percentage of that total microbiome. So the idea of the filtration is first, I didn't want to introduce enrichment to the stool sample so I don't create bias in term when I do sequencing. So it's kind of called direct sequencing of the stool samples. And the second aim was like, okay, how can I get more Campylobacter cells and less background noise from other bacteria? I mean, prior to apply it to the stool samples, I made like a cocktail of other bacteria and I filtered it with 0.65 micron syringe filters, which is a size a filter with a size that allows Campylobacter cells to pass through the filter and stops other bacteria, for example, Salmonella, E. coli, which is known to be a lot in the stool samples. So this gives Campylobacter more chances to be in the sample, and the other background noise bacteria less chances. And this kind of, when it's just when you think of it, if you have less bacteria, more Campylobacter, even with tiny amounts, but the percentage of the presence of Campylobacter is higher than one in the unfiltered sample. Our results show that increased the quality and the number of Campylobacter reads in the sequencing data, which like led to increased identification of species and typing on the and getting basically higher genome completeness of Campylobacter that allows us to do the identification and the characterization we want to do. So that's the whole idea of filtration. But I have to add that the filtration itself, it's laborious and it's not easy to do. So I think more improvements can be done in that.
Cool, thank you. So we have a lot of questions, and so the another one from Miriam Blanco. So Is it possible to characterize Campylobacter from wastewater samples?
So wastewater samples, I mean, it depends what method you are willing to use. And again, what I faced with Campylobacter, whatever project I'm working on, the first bit of it needs to be optimization. So you need to test the methods, validate them before even going to your project. I haven't worked on water or wastewater before, but imagining that it's not a rich media for Campylobacter, and as Campylobacter is quite sensitive to the atmospheric conditions because it requires microaerophilic conditions to grow and multiply, and oxygen basically is a poison to the bacteria. So, I mean, you have to test and optimize the methods.
Okay, great. So the another question, Sofia Ferraria, sorry, and can this method be extrapolated to the metagenomics in other bacteria?
Actually, that's an excellent question because part of my discussion in the paper, it's not only Campylobacter. I happen to be working on Campylobacter, but it can be applied to other pathogens. And for example, as a side project, I also worked with samples that were Salmonella positive, and we were able to at some point recover the genome of Salmonella and identify it at a certain level. But again, it needs to be tested, and because Campylobacter has a different genome size, other bacteria has a different genome size, and it depends what is the coverage you need and the depth of your sequencing. It's all those criteria that need to be taken in consideration to say yes, it can be applied. But in general, why not? I mean, Campylobacter is a fastidious organism. It's not an easy organism to work with, and it worked. So
Okay, thank you. And the other question from Mike Wanga. Thanks for the good talk. Were there any samples negative for PCR but positive with genomics?
that's actually a really good question because here we always, I mean, that's something I'm finding in my current project in the last slide I showed you. So in some samples that this is actually a discussion point in the paper itself because the traditional method which is now used in the diagnostic labs is the PCR assay, which is limited to only 3 species. And the reason why 3 species of Campylobacter, because 90% of Campylobacter infections are caused by the species Jejuni, and the rest of the infection is between other 2 species, coli and larvae. So and technically, you are losing the identification of other species because you have other species of Campylobacter. And this can be like a limitation for cases called unknown symptoms, and you could be missing other species. So when I tested that in my next step study, I would call it, and I tested like a group of people that were PCR negative for Campylobacter based on the screening from the diagnostic lab, 30 patients and 30 patients that are Campylobacter positive. In the Campylobacter negative ones, metagenomically, I detected Campylobacter reads in the. So using sequencing, you kind of conclude that it's positive for Campylobacter, but also for other species. And then here comes the question again, going back to the bioinformatics. What is the threshold? When do we say it's positive? How many reads we do need to have to say it's positive for Campylobacter or negative? Because that goes back also to the softwares that we are using, how sensitive they are they too sensitive, and those kind of things. This is a debate, and I'm afraid I don't have the answer for it now. So But we do find using sequencing that some PCR negative and culture negative have Campylobacter reads, but not enough to recover the genome.
Okay, thank you very much. Obviously, we have so many questions, but because of the time, so sorry we can't reply all of them now. And thank you all for your time today. And yeah, we hope you enjoyed the webinar and found it useful. And again, thank you for all your questions. As we mentioned also, and we haven't managed to answer your question live, we will get back to you via email next week with the answer. So in addition, we will email you a link to the webinar recording and the slide deck. And you can expect to receive it the week after. And when the webinar finished, a survey will pop up. We'd appreciate you taking the time to complete this, and as it helps ensure we make future webinars even more relevant to you and your research goals. And so there will be a link in the follow-up email for you to view a certificate of attendance if you need one. And this can be printed from your browser window. In the meantime, you can follow us on Twitter and LinkedIn to keep up to date with future webinars, offers, news. So yeah, we look forward to seeing you all the next webinar. Thank you very much. Have a lovely day.
Thank you, Sylvie, and thank you, everyone. And I'm happy to take questions on LinkedIn in case you have.
Thank you, Bilal, for your time. Thank you.
Thank you. Bye.
Bye-bye.